Hello and welcome back. This is part 2 of chapter 2, the chemistry and energy of life. Last lecture we talked about basic chemistry, such as what makes up an atom, and ionic, covalent, and hydrogen bonds. This chapter we will focus on some important biological molecules, such as lipids, proteins, and carbs. We'll end the chapter by talking about changes in chemical energy, and chemical reactions. So let's dive in, shall we? Molecules may have what are called functional groups. This is a small group of atoms that are attached to a molecule that give that molecule specific properties. These functional groups can determine the molecule's shape, reactivity, and polarity. Above you can see some examples of functional groups that may be attached to molecules. During this lecture, we will go over some important biological molecules. We will learn about their structure and functions. The biomolecules we will learn about are probably some that you've heard of. Proteins are formed by amino acids. Carbohydrates are formed by monosaccharides. Nucleic acids are formed by nucleotides. And lipids are formed by lipid monomers. But before we talk about them, we will learn about how these molecules are made. The biomolecules that we will talk about, and many molecules that we will learn about this semester, are usually made in two types of reactions: condensation and hydrolysis reactions. A condensation reaction are reactions where water is removed, and this links the monomers together. The monomers are molecules that are bound together. When linked together in long chains, they are called polymers. We'll see examples of monomers and polymers in a moment. A hydrolysis reaction is where the addition of water breaks the polymer into monomers. When learning this term, think of hydro meaning water and lysis, which means to break apart. On the top picture here is an example of both condensation and hydrolysis reactions. If you remove a water molecule, these two molecules, methanol and methyl ethanate, will be bound together. If you add water to this molecule, it will break apart into its original molecules. Here's a general example of a condensation reaction. You have two generic monomers that have a hydrogen and an OH group, also referred to as a hydroxyl group. By removing these atoms, you remove a water molecule, which brings these two monomers together, as you can see on the bottom. And here is an example of a hydrolysis reaction. Again, you have a generic polymer at the top. With the addition of water, it will break it into its monomers. Alright, with that out of the way, let's talk about some important biomolecules. First one we will discuss is carbohydrates. Carbohydrates is a source of energy within living things that can be readily transported. Also, it can be used as a structural molecule that helps give organisms their shape. Also, carbs can be used in cell response as recognition or signaling molecules, such as on the surface of a cell, which can help the immune response or to transfer other molecules into the cell. Monosaccharides are referred to as simple sugars. Two types that are important in biology are pentose, which have 5 carbons, and hexoses, which have 6 carbons. Two important pentoses are ribose and deoxyribose, which form the backbone of RNA and DNA, respectively. Hexoses include glucose, fructose, mannose, and galactose. Here are diagrams representing 5-carbon and 6-carbon carbohydrate monomers. Ribose and deoxyribose, which are pictured on top, have similar structure but differ by their functional group, which you can see highlighted in blue. Likewise, on the bottom, you have 6 carbon sugars. All of them have similar structures, but differ by a functional group. It may not seem like a big difference, but these functional groups can determine what properties these molecules will have. Monosaccharides are bonded together through condensation reactions. For example, by removing water from glucose and fructose, they are covalently bonded to form sucrose. This covalent bond is called a glycolic linkage. By linking 2 monosaccharides together, they form a disaccharide. An oligosaccharide contains several monosaccharides. They are often bonded to proteins and lipids on cell surfaces and used in cell signaling. Blood types get their specificity, such as A, B, or O, or positive blood, from oligosaccharide chains. You can see on the picture on the right of red blood cells. The oligosaccharides are attached in the chains, which help give the red blood cells their identity. Polysaccharides are larger than oligosaccharides and are large polymers. They can form chains of many individual monosaccharide subunits. These chains may be straight or branching, like starch, pictured above. Starch is just a large polymer full of glucose, while glycogen is highly branched and a main source of storage energy within mammals. Polysaccharides in long, complex chains are known as complex carbohydrates. Why does it matter if polysaccharides are branched or unbranched? On this slide we have several polysaccharides, which are made of glucose. Cellulose on the top is unbranched, has chemically stable links. Cellulose is an important structural polysaccharide found in plants, where it's found within the cell walls. Starch and glycogen, which are both made of glucose as well, they have different degrees of branching and are both readily used as energy sources, where cellulose is not used as an energy source. This slide demonstrates the degree of branching between starch and glycogen. Starch on the left, glycogen on the right. Starch has less branching, as seen on the top left, than glycogen. As stated before, cellulose does not have any branching within its structure, and is the main component of cell walls in plants. Cellulose is one of the most abundant carbon-containing molecules on Earth, which is attributed to its very stable nature. Here on the left is another picture of cellulose. Again, notice that there is no branching glucose coming off the main chain. This allows cellulose to form parallel chains that can bond together via hydrogen bonds, thus adding to its strength. Where the polymers that do have branching on it are not as strong structurally as cellulose. As stated before, complex carbohydrates are polysaccharides that form long, complex chains. Also, complex carbs take longer to digest than simple carbs, such as raw sugar. Also, complex carbohydrates tend to ask more complex questions, such as why are we here, and what is the meaning of life? Alright, here's our first progress check. Do your best to answer these questions. Please review the material. Also, it's always a good idea to read the book as well. if you have any questions or if something is unclear. After you let that material digest, move on to the next part of this lecture. Next important biological molecule we will learn about are lipids. Lipids are also referred to as hydrocarbons, which are molecules composed of hydrogen and carbon. The picture on the top illustrates a generic lipid. Note all the carbon and hydrogen atoms. Lipids are insoluble in water because of their many non-polar covalent bonds. There is energy in lipids' carbon-to-carbon hydrogen bonds. Lipids also play a structural role within cell membranes, as we will see later in this lecture. Fat, which is a lipid, can be used as an energy source or as thermal insulation within animal bodies. One type of lipid that we'll explore are known what are called triglycerides. These are also referred to as simple lipids. Fats are solid at room temperature and oils are liquid at room temperature. A lipid is solid or liquid depending on what type of bonds they have. Triglycerides have very little polarity and are extremely hydrophobic, meaning they don't dissolve in water. Triglycerides are composed of 3 fatty acids and 1 glycerol molecule. Fatty acids, represented by the black on this diagram, are non-polar hydrocarbon chains, which is attached to a carboxyl group. The glycerol, which is the red part of the molecule, has an alcohol and 3 hydroxyl groups. Triglyceride is synthesized using 3 condensation reactions. On the left in this diagram, we have a glycerol molecule and 3 fatty acids. With the removal of water in the condensation reaction, we get the formation of the triglyceride on the right. Lipids can be either saturated or unsaturated. What does this mean? Fatty acid chain in a lipid can vary in length and structure. Saturated fatty acids have single covalent bonds between all carbon atoms. They are said to be saturated with hydrogen. Unsaturated fatty acids have chains that have 1 or more double covalent bonds. This prevents the molecule from packing together tightly. As you can see in the top picture, this is a saturated fatty acid. All the carbons have single bonds with all the other atoms. The bottom picture is an example of an unsaturated fatty acid. There is a double bond between 2 carbons. This can make the fatty acid chain less dense since it will have less hydrogen. Unsaturated fatty acids do not pack tightly together. This gives them low melting points, which allow them to be liquid at room temperature. Here is another picture of a saturated fatty acid. Notice the structure on the left. All carbon atoms have single bonds with other carbon atoms. They have the largest number of hydrogens possible, making them saturated. Here's a diagram of an unsaturated fatty acid. It has two carbons with double bonds, which you can see highlighted with the yellow. Fatty acid needs only one double bond between carbons to be unsaturated. This fatty acid has two. Double bonds tend to make the fatty acid not be as straight as a saturated one. They will have a slight bend to them depending on the number of double bonds. Another important type of lipid are phospholipids. They have two fatty acids and a phosphate group bound to a glycerol group. Phosphate group has a negative charge, which makes this part of the molecule hydrophilic, or water-loving. Fatty acids and phospholipids are said to be amphithetic, meaning that they have a hydrophilic and a hydrophobic end. Here is a structural diagram of a phospholipid. Notice the phosphate group on the top, which is the hydrophilic end, and two fatty acids, which is the hydrophobic end. Within an aqueous environment, phospholipids will form a bilayer. The nonpolar hydrophobic tails pack together inward towards each other. The hydrophilic phosphate-containing heads face outward towards the water. Cell membranes have this type of phospholipid bilayer structure. Here's another diagram of the phospholipid bilayer. Remember, the hydrophilic phosphate heads will face the water, while the hydrophobic fatty acid tails will face away from the water. We will learn more about this in the cell membrane chapter. Alright, here's another progress check. Answer these questions and move on with the lecture. We are going to change gears a bit and talk about chemical reactions. All the molecules we have talked about, such as carbohydrates and lipids, will eventually be used within a chemical reaction. As we will see in later chapters, they can be metabolized to produce fuel for the body, or used in other functions. Chemical reactions occur when atoms have enough energy to combine or change bonding partners. The reaction here is showing that sucrose plus water will yield the products glucose and fructose. Quick review. What type of reaction is this? Key to the answer is that the water is splitting apart the sucrose. It is lysing it. You said hydrolysis. You are correct. Chemical reactions involve changes in energy. Energy in the chemical sense is defined as the capacity to do work, or the capacity to change. Within biochemical reactions, energy changes are related to changes to the properties of the molecules. We will explore this further. All forms of energy can be considered as potential energy or kinetic energy. Potential energy is stored energy. In the picture illustrated above, you have a biker at the top of the hill at rest. The object has stored energy due to gravity and sway. Once the bike is released and goes down the hill, that energy is now kinetic energy until the object comes to a stop. Kinetic energy is the energy of movement. This is the type of energy that does work, that makes things change. Within biology, think of a sugar molecule. It has energy. When it is not participating in any reactions, that energy is potential. It's being stored. And that sugar is being used in chemical reactions is now kinetic energy. It is changing and producing heat as well as other products, such as carbon dioxide. Energy can be converted from one form to another. Metabolism is the sum total of all chemical reactions occurring in a biological system at a given time. Metabolic reactions involve changes in energy. Energy is either stored in chemical bonds, such as after you're done eating. This is an example of potential energy. Or it can be released from those chemical bonds while you're exercising, which is an example of kinetic energy. There are two types of metabolic reactions. First one is called an anabolic reaction. This is when simple molecules are being linked together to form complex molecules. This type of reaction requires energy input, also referred to as inorganic reaction. Energy is captured in the chemical bonds that form. Anabolic reactions are illustrated in the top two pictures here. Over the course of the reaction, there will be more energy at the end within the final product than there was at the beginning. You can see your starting energy here, and at the end of the anabolic reaction, your molecule is going to have more energy because of the combining of the molecules. The second metabolic reaction is called catabolic reaction. This is a reaction that releases energy from a product. This type of reaction can also be referred to as exergonic or exothermic. Here, complex molecules are broken down into simpler ones. Energy that was stored in the chemical bonds are released. Catabolic reaction are illustrated by the bottom two diagrams here. Over the course of the reaction, the initial product releases energy as its bonds are broken. This will lead to the molecule being split up as the energy is released. Final products will have less energy than the initial product. Catabolic and anabolic reactions are often linked. The energy released in a catabolic reaction is often used to drive anabolic reactions. Think of when you burn fat tissue, energy is being released, which is the catabolic reaction. To produce muscle energy, molecules for your body, which would be an anabolic reaction. Transfer of energy from a catabolic reaction to an anabolic reaction isn't 100% efficient. The laws of thermodynamics apply to all matter and energy transformations. The first law states that energy is neither created or destroyed. It just changes from one form to another, such as potential to kinetic energy. Second law states that useful energy tends to decrease from one reaction to the next. This means that energy becomes unavailable for doing work when it's converted from one form to another. This diagram is illustrating energy over time. Each box is a different product after a reaction. At the first reaction, you have a lot of free energy, which is illustrated blue. As the energy is converted to another form, such as a molecule, some of that free energy becomes lost. Over time, as the energy is converted from one form to another, the unusable energy increases. Free energy decreases over time because no physical process or chemical is 100% efficient. Some of this energy is released as heat and becomes lost. The energy that is lost disperses as non-usable. This is why we must continually eat in order to replace this energy. As energy is being lost, entropy increases. Entropy is a measure of the disorder within a system. As a result of energy transformation, disorder tends to increase. Metabolism creates more disorder than the amount of order that is stored. For example, in order to gain 1 kilogram of body mass, an anabolic reaction requires about 10 kilograms of food. If metabolism was 100% efficient, you would gain all 10 kilograms. Because of this inefficiency, life requires a constant input of energy to maintain order, or better known as staying alive. As another example, about 10% of energy is lost between food web levels. This is why there are more plant eaters than there are top predators. A grizzly bear or an eagle require a lot of food energy to stay alive. Okay, here's the last progress check. Take a minute or two to answer the questions and enjoy some more ChemCat. That concludes Chapter 2. I know we went over a lot of material, so please review the chapter and let me know if you have any questions. I know chemistry isn't everyone's favorite subject. If you keep up with the lecture and review your notes, I know you can do it. ChemCat and I both believe in you. Thank you and have a good day.